Good morning to you all. It's a privilege to be here. I found Christina's part really interesting and saying because it talks about how we georeference and with this talk, I'm going to really talk about what is the importance of what we do, why we do it, and what research. I'll do it in research is in a general view, why we do research on spatial data. Because I've had a whole career at the Natural History Museum now, many years too long, but I've always shown to be important to me to try and unlock, I call it the spatial wealth of collections. Now, the geographical side of collections, the label, where a collection is collected, is fundamentally important. And it has always been an area to me that has been not so much ignored, but it's always been taken to be the last thing that we do. Now, if we use collections, and if we use natural history collections, they have a massive input outside the area of collections, and they can really influence future studies. So this is where I'm going to come from. So let's go to the first slide. I always like to start off a talk and saying this: a map is worth 1,000 words. And we all know that. There's so many maps that we see, whether it's COVID before, where we're looking at COVID maps from Patrick Vallance, which we probably don't really want to remember too much about. But remember, we saw maps. Everything we do is about maps. But a map can only be a map if it has a latitude and longitude, which is georeferenced data applied to it. Now, our life is reminded, whether it's Google Maps, whether it's on this, whether it's on that, everything we do is reliant on a geographical latitude and longitude, where we are, how we interpret it. If you can go to the next slide, please. So why do we georeference? We hold vast amounts of data, both in the Natural History Museum, both in NatSCA organizations. And I estimate that if the museum, the Natural History Museum, has 80 million specimens, which it was 70 million when I started, and it's now 80 million, so it's about that. We don't have 80 million sites that they've been collected from. From all the work that I've done so far, from different georeferencing projects, we probably have about 3.5 million sites, both across the world and also extraterrestrial, some of them on Mars. So 3.5 million, and it varies again what collections you have. The experience that I have had, certain sites have many collections that are collected from. And a good example is things like Lepidoptera, insects fusing collection devices there. Obviously, sometimes it's just one site. But if you go to things like orchids, I think the Natural History Museum has about, correct me if I'm wrong, but call it 15,000 specimens of orchids. And I remember when we georeferenced them, we had about the 14,000 individual sites within the UK that have been collected. So that's quite a large number of sites. But if we looked at things like Lepidoptera, those we've done across the world, I think there was about 200,000 Lepidoptera specimens within the NHM collections in the UK, and I think they ended up about 10,000, 11,000 sites that represented the whole collections. So what I'm trying to say is we haven't got 80 million specimens, 80 million sites that we need georeferencing. We probably have no more in the Natural History Museum than probably about 3.5 million, of which many will have been georeferenced by other institutions. And this is where we have to talk to each other. And this is where we have to share information, because there is no point georeferencing some data that has already been georeferenced somewhere. You might well want to check it, but it's very important that you don't overdo it by georeferencing everything. Again, we can save time by using information and amalgamating it. So I always think, again, that we have a special position, both in the Natural History Museum, both in NatSCA organisations, to open up a huge spatial resource. Now, the importance of this is, how do we use this data in the future? How can we use it? How can we open it to different audiences? Now, I always use an example of species distribution. We can use species distributions across different institutions across the world, right? Put them together and we can add resolution to climate models. We can do the historical work. We can look back in the past to find out where distributions have changed over time. We can also, very importantly, look at the error that we have or the precision of the data. That is one area that is fundamentally important, and that is one area why it's fundamental for georeferencing to have a level of precision that can be used within models and models for looking at climate change. For instance, as an example, if I want to look at a distribution and put a species distribution and compare it to a soil type, a geology type, a forest type, an ecological zone, and I produce a very nice model that predicts that these species could be in A, B, different areas, you might find that that model doesn't fit exactly how I want it to do. You might find thinking, well, okay, why doesn't this model fit? So when you have a precision or when you have an accuracy level within your data that you have painstakingly georeferenced, you can then start to take data out. Look at where actually we've only got country records for this specific species. This is why we're getting spurious data in these areas. So that is one of the major importances of why a level of precision is needed for georeferencing, so we can use it within different climate models, species models, whole kinds of raft of extra research data for the future. Finally, from this slide, here's a bit of work that I used and that I did in a long time based on Christina's work. A map is showing everything. Here we go. We've got a hotspot map. You can guess that the redder areas is where the Natural History Museum has more Chinese types. And you can see the dots in Sichuan in the center, Yangon. And you can also see, if you look at it, one spot or the larger red dots account for 144 type specimens that the Natural History Museum has. What I'm saying is the power of a map, the power of finding out a georeference can show you a great deal. Let's go to the next slide, please. So when you georeference, as Christina has kindly shown, it's fundamentally important that we have a set of guidelines, develop a set of guidelines. If you're starting off, it would be the one thing that I would say is the most important thing. What you must have is a set of guidelines which you can get from other institutions, you can collaborate with other institutions, or you can merge their ideas. These are not unique in any way. I developed these over time by using, I think originally, Manus and HerbNet and Ornis data. And what we've done is we want to ensure that people, when they georeference or if we use automation programs or anything like this, with the georeferencing process is repeatable. So what I do will be the same as perhaps Christina will do, perhaps Hilary will do, who will talk later. So if they're georeferencing, we'll get to the same result. Or if we get to the same, a relatively same result. Now, to do this, you must have a set of guidelines that get your teams of people to georeference a river meander, if that's where you've collected, a bridge, using a coordinate system. What do you do with a coordinate system, or if the data, the projection is in a UTM? What do you record it as? Now, these guidelines here are, which we will make available later, and they're literally there's pages and pages of these, give you how you would georeference a junction of a road, for instance. It will give you, if you look on it, a procedure, and it will give you a determining extent. This is the error, precision, whatever you want to call it, that is very useful for data to be used externally to the museums and NatSCA organs in, let's call it scientific, pure scientific research is very important. But if you look at this, how do you deal with things like nearby? How do you deal with a street address? How do you deal with a river mouth, the mouth of a river? Here we have a set of guidelines which you will not always agree with, but what it gives you is a way of repeating the same georeference for a different specimen. And in that way, we have it makes it very important that someone in the future, which will probably in 50 years' time or 40 years' time, from some CMS will turn around and say, right, I understand why they georeference this way. Do I agree with it? Maybe not, but I understand why. So I can actually look at this data and say, well, actually, they use this old type of GPS. Oh yeah, this was collected in the 1980s. Oh, it's extent was probably only 40 meters, 50 meters, 30 meters, or they used Landsat data, old Landsat data, which is a satellite image, and its resolution was 30 meters. Oh, they couldn't get any more accurate than that. So this is where it gives us a guideline how we should be doing things. And it's fundamental, as far as I'm concerned, that in any georeferencing project that you do, you have a set of guidelines, which is not a transient, it's not a thing that is static. You will find, as I find all the time, a different label comes up, a different transcription comes up, and then we have to add a method of dealing with how we should georeference it. And just as I said, just really when I think it's over, and I don't need to add any others, it comes up. So if I say, even though you can't really see a great deal in this guidelines, having a set of guidelines of how you should deal with an event, how you would go out and georeference a species that has perhaps been collected from a bay, the Bay of, if you, for instance, I don't know, the Bay of Biscay or something. There is an example here of how we should do that. So this is fundamentally important, and this should be followed. And this basically allows us to be repeatable. And this is where the value of the data can be used externally to perhaps what it was collected for in scientific research. And you'll notice on there, I've put a measure of precision of a georeference is very important. If nothing else comes out of this talk, I would like you to go away and say the precision that you've got is incredibly important. I'm not talking about an error, it's a precision. It gives you guidelines for how you can look at this information. Next slide, please. Making georeference easier. If we all had enough money, we'd all want teams of people going away and georeferencing, following our guidelines and going away and georeferencing. That's not the case. So what we've got to do is make life easy for everybody. Now, there's degrees of automation. We should follow the automated techniques that are already available. Google Maps is a very useful thing. GeoNames, I think GeoPick was mentioned earlier on. If we can automate the georeferencing of individual specimens that usually are generally easy to georeference, repeatable, then you should do it, because it will save you great deals of time and probably deal with a significant amount of different people's collections. And the important thing again about that, it then allows the team of people that are georeferencing the time to actually work on the very difficult ones, the ones that were collected by a bearing from an expedition when you only know the start and the end, but they know that they followed a river. Time to spend looking at geographical changes over time, historical name places over time. That is where you need to spend the effort. So really, if you should make use of automation. And there are a number of systems that are available, which will make life a lot easier for you. And interesting, I did something like this, looking at kestrels of all places in Madagascar only a few weeks ago, with a researcher in the museum. And literally, he'd got the data out of our CMS, pulled it all out, couldn't georeference, couldn't find it. I ran it through an automation program, which I knew you used, and it was from Madagascar, and it picked up, I think, 90% of them with an accuracy of probably between 90 and 98%, which basically meant it was absolutely spot on for the data that he'd got. And it picked them all out, and literally took me 3 minutes for thousands of records. So that is the beauty of it, where it works. Now, it will not work when, for instance, you're dealing with historical data or things that are changing. You've got to have expertise. And Christina did mention this, and I will reiterate this. There is a great benefit for having local knowledge, language knowledge. Just there, I can understand different languages, different things, but sometimes the context is very different if you are an indigenous or you're a local people. And again, a lot of data that I've collected in South America and different things, I tend to try to get georeferenced by local people who know all the different names and who know all the different derivatives of names that they've got, and also know far more than I do. So leveraging their information, because they've got stacks of information, leaving their knowledge is very important. And I would always push forward is saying this very, very important that you include, if you can, local knowledge or experienced knowledge, people who have experience of different geographical areas. I've put also in this slide, I've put GISs, ArcGIS Pro, QGIS, they've all got georeferencing tools that are all very useful. And again, Google Earth does as well, Google Maps, all of these things they should be part of your approach, because certain systems work very well for certain different geographical areas. I've had certain automation programs which work very well for North America and not so well for Europe, and vice versa. And they get different answers and things like that. So again, having the knowledge, having the experience to go through this type of data is very good. AI, it's a buzzword, of course, at the moment, and everything's AI, but it is incredibly, some of the language learning facilities is incredibly useful. And again, I see this developing much more in the future, and the ability to learn, the ability will fundamentally help with the georeferencing of named places, or say named users. All of this is going to be particularly important in the future. Now, is it there yet? Not entirely, but it will be, I'm pretty sure. And it will again work in that, I call it that automation, that AI kind of phase, where perhaps you can have collections that you might well be able to pick up 80% of your, let's call it specimens, in one foul swoop. You've still got to check them and you go through and make sure that they are of the standard that you want. But it's a growing area, and I only see it helping us to develop and georeference the collections. Because I still have this dream in the Natural History Museum that when I retire in many, many years' time, that all of our collections will be georeferenced, and I'll just be able to Google, put them all up, pull up anything that I want, knowing that it's georeferenced. It might be a bit of a tall order. Accuracy, again, I've mentioned before, accuracy, precision, repeatability. That is where the guidelines come into things. That is fundamentally important in georeferencing projects. And also, I have passing it, don't reinvent the wheel. Share data where possible. That is the important bit. There is no point really that georeferencing collections that Missouri have already georeferenced. There is no point me georeferencing certain things that Q or RBGE or whoever have georeference. If it is done to a common set of standards and to a common level of precision, like that, we should use their data and our data and amalgamate it to help me. And this is where the benefits of things like portals, pulling data together, standards that we were talking about, Darwin Core, all of this allows us to do that. So it's very, very important. Let's go to the next slide, please. So what can you do with this type of data and how, and let me start to show you a project that I'm working on. I've been working, well, people have been working for me on georeferencing the botanical types at the NatSCA Museum. And this initially is just to show you, this is a hotspot map. Another simple map shows you what it needs to do. I'm not showing you the number of specimens or the number of sites. It's just a hotspot map of the data that was pulled out of the CMS about 4 or 5 years ago. So you can see instantly we assume we've got more data, so more sites for the types that were collected in Southeast Asia, the UK, and Central America. Probably, if you know the NatSCA Museum, you'd expect that. But again, it spreads down to South America a little bit. Now, but that was the initial data. And remember, this data was probably based on data that said Germany, UK, Barbados. That's all we said. But there's there was loads more information because that was the level of our georeferencing of the types, of the botanical types at that time. Most of it was georeferenced at that time 5 years ago, was literally myself I think it was myself producing a number of centroids for different countries and then linking the country field saying, right, okay, this collection was collected in Bangladesh, or sorry, not Bangladesh, India, or something like that, and the georeference of this centroid is this, and that was the level of our georeferencing. So that wasn't good enough for our types, and it's not good enough for what we want to go forward. So we've now been georeferencing. Next slide. So what have we been doing? So 137,000 specimens on the top left is how many botanical types roughly that we have in the Natural History Museum. So they are presently being georeferenced using the methods that I talked about before. And if you look on the right, there's Australia, nice map of Australia. You see the limited number of dots. Those were the original sets of data that came out of the CMS 5 years ago. These are the types which represented, I think, about 8 or 9,000 types that have been collected around Australia. So not a great deal, really. So there. So now we started to georeference. If you look a little bit more, you'll notice by the Bay of Carpathia, I think, which is the red area located in northern Australia, you'll start to see there's more now. And you'll notice the structure around the coast, Tasmania, you think, we're starting to, Sydney or whatever, you can starting to see there is a range. Now, if you look to the furthest left at the bottom, there's this processing going on. Yeah. Now you can see there's a lot more. But this is where now the sites that we've developed, there's even more now, but you'll now see that we've got plenty of sites. The information was there. We just hadn't georeferenced them. And that took a lot of time. But if interestingly enough, the red dots there are Robert Brown's collections. So you can see, you suddenly get a story. You can actually pull out individual collectors. Where did they collect? How then? You can then start to study, map their locations, where they went, even the time-wise. You start to really pull together a great story, which is one, very good for research, but also two, because you bring in other audiences of people that are fundamentally interested in where the data was collected. Adding a little bit more to that slide, and there a lot of the slides that actually Liz DeFell had to georeference were things like old mine sites, old Aboriginal monuments and things like that. The names have changed and things like that. Now, these places were, again, we couldn't find them, but what we had to do, or what Liz had to do, was spend a great deal of time working through the archives, working through the labels, looking at the expedition logs, looking at the logs, the journals, the individual journals, because the information is there and it's incredibly valuable. But I think you would have found initially that probably, if you look to the top right, there's probably about 20 dots would represent all the types that the Natural History Museum has, botanical types for Australia, which was not correct. Anyway, next slide, please. So, South African types, interestingly enough, now this is, again, South Africa. You'll see, and again, this one, the yellow dots. That's the yellow dots you can see initially was what came out of the CMS. Now, the red dots are where we are now. As one can imagine, these georeferences, again, a lot of it follows the colonial, the British Empire. So again, there's a story there. And you can actually see you can see how, for instance, there is a great deal of detail there. There's a great deal of geographical information that was hidden and unavailable to researchers, or perhaps only known to a few curators. So what I'm trying to say again is value is added. And again, each one of those sites is not just specific to a type, it could be specific to other collections. So again, these type sites are particularly important because you can then link a specimen to perhaps it was collected at that type, but of that site, but isn't a type. So you've got here 35 sites originally in South Africa, as an example, now equates to 4,234 different type sites that the Natural History Museum holds. So a great deal of information is there. And again, a great deal of information can be georeferenced and added to. Next slide, please. Okay, so we're using this data, and again, I'm talking about georeferencing, and I'll give you an examples of just how we can use it away from the field, perhaps, of NatSCA and things like that into the research. Simple, here you go. This is a georeferenced satellite imagery, right? And the word georeference, instantly, that image on its own would come as a series of pixels with no real link to the Earth's surface. It is now georeferenced. Underneath there is a load of sites that people have gone out with GPSs, looked at, and actually tied it to that image. That image is of actually a particular area in Argentina, and it's actually looking over from the Andes over to a national park called the Nahuapi National Park, which I was doing vegetation classifications on. But what it's trying to say is georeferenced information, again, fundamentally important because it opens up other avenues where we can start to use vegetation data, biodiversity data, collecting all different forms and tie in the importance of collections. Next slide, please. So what can we do from this type of data? Again, vegetation. This is a vegetation map. This is of a country in Belize and Guatemala, actually, and it's a vegetation thing. All of this is based off satellite imagery, collections going into these areas, collecting specimens, getting a coordinate, making sure that we actually can tie this to produce a vegetation map, which can be used and is used with other forms of data to tie in what's going on the ground, assess biodiversity, ecological change, tie it to geology. Again, you can produce all things, again, georeferenced. Next slide, please. And again, what can we do from some things of this data? This is this is actually carbon, soil carbon, collected, georeferenced from those little dots in the middle. This is a selection of universal translocators, or UTM, coordinates that I collected in the field many, many years ago, right? Which I collected in UTMs because it was easier for me to do that in Central America and there. And literally from that, I then produced this model of carbon data that has been organic carbon data collected from soil samples, which were analyzed in the lab, but based on a coordinate for each one of those points. And then that produces this nice little model, which basically says, as I would expect on the bottom right, the higher ground, that's the higher ground, which actually corresponds to a deciduous tree, a set of deciduous trees down to the lower ground to the left, which is more evergreen. So obviously you get more leaf fall in the deciduous areas, so you get a higher level of carbon within the soil. Shows you very nicely. Next slide, please. And again, now, this is of a country, Belize, as an example, but again, this is actually a model of a distribution of Charté, which is a Chamaedorea, the different Chamaedoreas across the border, and it's showing the variation of Chamaedoreas and Chamaedoreas across from Guatemala to the left, Belize to the right, to the Caribbean coast. And obviously it varies and things like that. There's high concentrations on the bottom on the left. And again, this is based on georeference data. It was based on data that was collected in the field, locations of specific Charté plants. We have the coordinates, we have the specimens within the collections. We've gone there, and literally it's been based on georeference data collected in a standardized way, which then allows us to model and produce a distributional variation of a specific plant species, which is easy to understand because we all know if you look at it, if you look at the key, the higher areas, I think it's more abundant areas are the browner areas, the lighter areas are where it's not abundant, simply. Next slide, please. Now this one, you might not think of it, but it shows another way of using georeference data. Imagine this. This is a Neanderthal tooth, and this is cadmium levels on that tooth. Each one of those dots, each one that was there, is actually a georeference neoblation on a neoblation technique on that tooth. So from that, we produced a map of a tooth structure. That's actually then modeled the variation of, I think it's cadmium and strontium on that tooth across there. And the way this works is basically you can then map neonatal variations and diet from the tooth by looking at where I think the yellow bit right in the middle is I think it's the neonatal line. But what I'm trying to say is scale. It doesn't have to be the world scale. It doesn't have to be, it can be anything. This is millimeters. But you can still use georeference data if you use the right collections and look at things in a different scale. Next slide, please. So again, here, more typical things, georeference data we've collected. This is just showing it's a black and white image. I don't know why I decided to do it black and white, but I did it black and white. I thought it might show a little bit more detail or be a little bit more arty, but it hasn't quite worked out. But anyway, what it does show is the darker areas are the higher ground and the lighter areas are the lower ground. And there are two different plots that we've collected from, and again, they are a comparison based on georeference data. Next slide, please. Now you've seen this probably from Christine. Again, this was the type specimens from China. Again, a simple map, georectified, shows them easily. We can already understand them. Very good for selling. This type of thing is very good for selling these projects to perhaps policymakers, people who have money, saying this is why it's important, because a map speaks widely. It's more than just saying, oh, we've got lots of collections in China. Yeah, you can even in this vague map, you can still see them. You can see in Taiwan, the museum has a great deal in the same. There you go. Next slide, please. So when you're georeferencing also, again, you've got this type of thing. These are just centroids that have been calculated for ISO countries and ISO class 1 departments and standardization. Now, again, if you have specimens that have just been collected from Indiana, you've already got that information. You can just literally link it to, very easy to do, very simple to do, and that will account for a great deal of your collections and actually give you clear wins very, very quickly. And it's always worth going through your collections that you have, going through, making sure you can get a large clear win very quickly. Next slide, please. Right. Now this, when it comes, this is going to show you really how I can use and how I have used georeference data in a specific project that I've had. Now, the point about this is the context, perhaps not the detail, but you'll see how a project can link together from a global scale to a much more micro scale based on one thing, the georeference. Next slide, please. Good, it worked. So you have a nice, again, for people were talking about earlier on, different projections and different things. This is a very skewed projection that I decided to produce because it shows North and South America for what I wanted to do. And again, one person mentioned about that, looking at projections and collecting data in different coordinate systems is fine as long as you make the decision of what you are going to display it as. In the museum, we do WGS 1984, latitude and longitude, decimal degrees. That's our default. But we can put anything into it. We can have UTMs, we can put polars, we can put anything into it. We just translate it. And that's relatively easy, at least for me, to work out what they are. Next slide, please. So here we go. This is an area actually in Belize. It's a tropical forest. The brown area here is, the brown area is a metamorphic plug, ex-volcanic plug, and the green areas, as you can imagine, is tropical forest of deciduous stroke evergreen varieties. And to the left, which you can see is more incurred there, that's where on the bottom left that's Guatemala. So you can see incursions, whatever, and you can see land practices that have changed. So again, a georeferenced image. This has been pulled out. It links into what I want to do. Next slide, please. So I've moved in a little bit more. This is a plot, and actually that plot, that image underneath, which is again georectified, is there. That is a 1 hectare plot. So you're starting to see a great deal. It's unprocessed data, raw data of actually tree canopies in this point. But you'll see that there's a number of white dots, which is my specimens, and there's a set of red dots in the middle, which is a grid that I, again, set up, coordinates, georeferenced, made sure it's specific. Actually, and again, this is in UTMs, Universal Translocator. So I've got the data, and again, the important bit is georeference. Next slide, please. There you go. There's that setup data again. This is actually, and this is the data and the grid. I decided to make things easier for myself. I've got a random. I set up a stratified grid within this, and these are 20 meters. 20 meter? No, 10 meters. I made a mistake. 10-meter intervals. Next slide, please. So what can I produce on this? Again, ISP means this is not carbon. This is not organ. This is where I've collected data at those sites, and I've produced a model which was based in this case on a Kriging method and a whole number of different methods there. But it uses spatial data, and the importance about this is having spatial, geographical, georeferenced data. And actually what it's displaying is not important here, but for the sake of this, I'm going to say that this is organic carbon. And again, organic carbon on the bottom, on the top right, bottom right, top of hillside, deciduous tree, bottom is evergreens, you will probably get less or more. I'm not going to say you get less leaf fall, but it influences the soils, influences whatever the influences the soils, the development, and everything like that. So we get lots of leaf fall, and that obviously influences the carbon at different types of the air. So again, what can we produce from that? The georeference data. Next data. Next slide, please. So again, okay, so let's look at it in a different way. That georeference data, I'm suddenly going, well, actually, no, I can now produce a surface. I can give it a nice way of looking at things. So I can actually look at where the hillside. So again, this is a digital elevation model. Again, produced from georeference data, pulled together, linked together. So I can actually look at where the higher levels of carbon are. And as I said before, now you've got the interpretation of things. You can see it's on a hillside. You can see, start to get an idea of how things fit together. And what I'm really trying to show there is the value of georeferenced data, whether it's a specimen, whether it's a soil sample, whether it's the location array. If we have a standardized method doing things, we open up avenues to use collections in general, the great wealth, I'll call it the spatial wealth that we hold within Natural History Museum, NatSCA institutions, which in some senses is unavailable to a lot of people. But if we do things, if we georeference them, I think the world literally will really, it's will open up because we can suddenly start to do this, whether it's genome analysis, whether it's there. The point is, is the spatial side of georeferencing. Next slide, please. Okay, so I said this is a shorter talk, but what I've been trying to get across to you is the importance of georeferencing. The standards that you use is fundamentally important. How we pull it all together and I'm giving you examples of what you can use it for. There's a multitude of others, and I really hope that if you're looking at georeference projects in the future, you look at what you can produce, because it is a really underused spatial resource that we can use right through science in general. And I'll say the final comment will be, a map is worth 1,000 words. Thank you.
Thank you so much, Malcolm. That was amazing, seeing all the stuff you can do with it if you've got the data. It's amazing. And it looks really pretty as well. We are running a little bit over time. So if you have questions for Malcolm, I might get most of you to hold on to them. There was one question that was asked during your talk, Malcolm, which was about whether you could recommend a good program or programs for modeling data.
Depends what you that's a bit of a nebula. Okay, it really depends on what you're modeling for there. I think that person would have to contact me directly and say, what are you trying to do? And I could give you an answer saying this is possibly the way that I might do it. Because again, it is unfortunately, it's like saying, what car would you buy?
Yeah, exactly.
Okay, it's a legitimate question. It's a legitimate, but it depends on the purpose of what you're trying to do. That's not me jumping out of the answer, but it's just a case of I can't really give an answer without knowing more.
Yeah, in touch with you after the talk. I'm sure there'll be, if you'd be happy for your email to be shared, I can send it on to her. There's another question here about the increasing precision over time as georeferencing improves. It's fascinating to see, but presumably only possible if you keep older versions of the data instead of overwriting it. Do you still have the older, less precise data, or just the maps produced from them?
The answer is yes, we do have it. You should never get rid of the older data because there is value within that, and you do need to see the iterations of how things change, because in the future you might find that there is an iteration of change that is wrong. or
Yeah.
someone else has a different interpretation of. So yes, and I think that's very similar. It's a very similar kind of questions. For instance, when you're doing like a botanical determination,
Mhm.
you want to know what happened before. You
Yeah.
want to know what, even if the determination has changed, you want to know why it changed. So you need to know the history behind it. So it's exactly the same here. You do need to know how the decision has improved, how things. So yes, you do.
Brilliant. Thanks so much, Malcolm.